This first activity is where we connect everyday observations to real scientific questions. We're going to start with something very simple, plants and their leaves, and use that to build a testable hypothesis that we can actually evaluate with data. The scientific method always starts with observation. You notice something about the world, and that leads to a question. Then you test that question in a way where only one variable changes. That way, if you see a difference, you can actually connect it to what you tested. Before scientists ever touch an experiment, they do their homework. They read, they think, and they figure out what question they want to ask and why it's worth asking. That's what becomes the introduction section of a scientific paper, the background, the context, and the purpose of the study. The introduction has to exist before the experiment because it explains why the experiment should even be done. But after the results are in, scientists often go back and polish it, tightening the wording and making the connections clearer based on what they actually found. A hypothesis is not just a guess, it's a testable statement. It forces you to be clear about what you're changing or comparing and what you're measuring to see if there's a difference. A really important point. In science, being wrong is not a failure. If your hypothesis isn't supported, you still learn something real about the system. Sometimes those results are even more interesting than the ones you expected. To build a good hypothesis, you have to be clear about your variables. The independent variable is what you're changing or comparing. The dependent variable is what you're measuring to see if there's an effect. In the fertilizer example, our question is what amount of fertilizer produces the most growth in native sunflowers? We're not asking about all plants. We're asking about one specific species. The thing we manipulate is the amount of fertilizer. The thing we measure is growth. Everything else has to stay the same. A hypothesis is typically written as a statement that includes the independent and dependent variable or a descriptor of the variable. Essentially, this is the answer to your research question that has not yet been tested. For the fertilizer example, this could be sunflowers that receive 10 grams of fertilizer will grow taller than those that receive less, or increasing the amount of fertilizer will increase the height of native Florida sunflowers. I've highlighted the independent variable in yellow and the dependent variable in blue. In science, we don't start by assuming our idea is correct. We start with the null hypothesis, the idea that nothing is different and nothing changed. Then we use data to see if that assumption holds up or not. If the data show strong differences, we reject the null hypothesis and support the research hypothesis. If the data don't show meaningful differences, we fail to reject the null. Notice we don't say prove. We talk about evidence and support. Most null hypotheses follow this exact pattern. The independent variable does not affect the dependent variable, or there is no difference between the groups. It's always a statement of no effect or no difference. This is what a proper null hypothesis would look like using our fertilizer example. It clearly states that the treatment, the fertilizer amount, does not change the outcome, which is plant height. Now let's bring this back to our actual experiment. These three simple observations are what guide this entire lab. That plants have leaves, that leaves on the same plant are usually similar in size, and that leaves from a different species often differ. They're things you already know, but we're going to turn them into a testable scientific question. I will show an example from measuring star anise, golden dewdrop, and snowbush, but you will need to choose leaves from plants near you. In this experiment, species is what we're comparing, so that's our independent variable. What we're measuring is leaf size, specifically the length and width to come up with the leaf area. So that is our dependent variable. Before we even write a hypothesis, we need a clear, testable research question. For this lab, the question is simple and very focused. Does plant species affect mean leaf area? Or said another way, is there a difference in mean leaf area among species A, B, and C? This question works because we can actually answer it with data. We can measure leaf area, we can compare species, and we can use statistics to decide whether any differences we see are meaningful or just due to random variation. These are the two formal hypotheses for this lab. The research hypothesis is the mean leaf area differs among the three species. This says there is a difference. The null hypothesis says there isn't. There is no difference in mean leaf area among the three species. Your data and statistics will tell you which one is better supported. These questions are meant to get you thinking like a scientist, not just repeating definitions. Make sure you answer these in your lab notebook. For the first one, remember that leaf area is just one way to describe size. Scientists can measure things like leaf length, leaf width, leaf mass, thickness, or even things like chlorophyll content. The point is to realize that choosing a measurement is a scientific decision, not an automatic one, and different measurements answer slightly different questions. For the second question, I'm intentionally giving you a different experiment so you can practice writing hypotheses, not just copying ours. The research question is about nitrogen fertilizer and bean plant growth rate. Your job is to turn that into a clear, testable research hypothesis, something that makes a specific prediction about what you expect to happen. There isn't only one correct wording, but good answers will clearly connect the independent variable, fertilizer concentration, to the dependent variable, plant growth rate, and make a prediction about the relationship between them. For the third question, remember that the null hypothesis is the baseline assumption. It represents the idea that nothing is different and nothing changed, and we use data to see whether that assumption holds up or not. That's what keeps us from starting with our conclusion already decided. And for the last question, think like you're actually writing the introduction section of a paper. You'd probably look up information about the species you're studying, what leaf area is related to in plants, how leaf size connects to things like light capture or water use, and whether previous studies have shown differences among species. That background is what justifies why this experiment is worth doing in the first place. The big picture from this activity is that science doesn't start with data. It starts with noticing things about the world and asking good questions about them. Those observations lead to a research question, and that question leads to a hypothesis that makes a clear, testable prediction. You also saw how important it is to be clear about variables, what you're comparing or changing, and what you're measuring in response. And you learned that the null hypothesis is always the baseline assumption of no difference, which keeps us from jumping to conclusions before we look at the data. Finally, this is where the introduction of a real paper comes from. Background research and observations that explain why this experiment is even worth doing in the first place.